Hello everyone, my name is Ana García from Hospital Sant Joan in Barcelona, and I am going to describe our JAIN paper entitled Humans with Inherited MyD88 and IRAK-4 deficiencies are predisposed to hypoxemic COVID-19 pneumonia. TLR7 is an endosomal double-strand RNA sensor that is dependent on MyD88 and IRAK-4 adapters to recognize SARS-CoV-2 and enhance the production of type 1 and type 3 interferon. These interferons will control lung damage secondary to SARS-CoV-2 replication. Thus, in cases where there is a MyD88 or IRAK-4 deficiencies leading to a defect in the interferon production, we hypothesize that there is a higher risk of severe COVID-19. In order to test this hypothesis, we described 22 patients with MyD88 or IRAK-4 deficiencies who were infected by SARS-CoV-2 before being vaccinated. There were 18 males and mean age was 10.9 years old. The analysis of other known genetic or immunological risk factors associated to severe COVID-19 was negative. Surprisingly, for their young age, we found that 16 of 22 patients had COVID-19 pneumonia in our cohort, of which 10 were hypoxemic and 1 of them died. Next, we analyzed the impact of risk factors associated to COVID-19 severity. First, regarding viral variants, severity was higher with variants that were dominant before the end of 2021, and none of the patients infected by the Omicron variants suffered from pneumonia. Regarding age, all patients older than 8 years developed pneumonia, whereas 60% of patients under the age of 8 years had mild infections. By contrast, sex showed no impact in COVID-19 severity. When we compared our cohort to an age-matched general population cohort infected by SARS-CoV-2, the risk of hospitalization, invasive mechanical ventilation, and death was higher in the MyD88 IRAK-4 deficient patients. For instance, the risk of invasive mechanical ventilation in MyD88 and IRAK-4 deficient patients was 70 times greater compared with the general population of the same age. In summary, our study shows that humans with MyD88 or IRAK-4 deficiencies are at a greater risk of COVID-19 hypoxemic pneumonia compared to general population regardless of age. The mechanism of disease in patients with MyD88 or IRAK-4 deficiencies probably involves an impairment of the TLR7 mediated sensing of the virus by plasmacytoid dendritic cells, as has been demonstrated ex vivo. In addition, we found that several patients in our cohort had had other previous viral infections that were severe. So far, that this investigation breaks the paradigm of the selective susceptibility to bacterial infections in MyD88 and IRAK-4 deficiencies, making paths for future investigations in this unexplored area. We hope this summary will encourage you to read our paper that has been done thanks to the international collaborators, patients, and their families. Thank you very much.